Hi everyone, so today we are going to talk about two projects. The first one is surface modifications and the second one is trabecular metal technology. And that's why we're here in the MicroCity lab. We have few equipments here. This is a MicroCity out here. We have goniometer and then we have a digital microscope out there to study surface and trabecular metal. And now I'll introduce you to Zephyr, who is going to talk about surface modifications.
Hello, Mr. Surgeon. Nice to meet you. So first I'll be telling you about the surface modification project. Here we have two main aims. The first one is to develop an antimicrobial surface. So you can see here, this is a trauma nail. This is for the femur, and here's one for the tibia. This might be an application that we look at for an antimicrobial surface because of the high infection rate and traumatic events. Second aim that we have is to develop a surface that's going to have better implant fixation and long-term osseointegration. That we'd be looking at like a dental implant, as seen here, because you want to have really good bone integration. So first thing we do when we get a new surface in is we want to characterize what does that surface look like, what are the elements on the surface. So here you can see a perspective surface. Here's a SEM image, and then we also look at energy dispersive X-rays spectroscopy to look at a color map, which will show us the elements on that surface. So you can see the bulk titanium in red, and then you can see the perspective surface in turquoise. So another thing we look at is the cross-section of a surface. Here you can see the bulk titanium of our implant, and then you can actually see the integrated surface. And with this, you can have an elemental analysis of the elements in the perspective surface. So the next thing we want to look at is hydrophilicity. Is this surface hydrophilic? Because you have better cell attachment with a hydrophilic surface compared to a hydrophobic surface. Here you can see that with a hydrophobic surface, cells are not attaching as well, water's not is beating up on that surface. With a hydrophilic surface, the water is going to spread across that surface, and therefore you're going to get better cell attachment. And I will actually show you a demonstration of that in a minute. So then we want to start doing in vivo or in vitro assessments. Some things we look at are cell attachment. Here you can see nuclear staining DAPI and F-actin stained by tritzy conjugated phalloidin. So this way we can look at the cell morphology, make sure the cells are attaching well to the surface, and we can also assess cell proliferation and cell attachment quantitatively using an MTS assay. Another thing we want to look at is to ensure that cells are differentiating on that surface. So when we're looking at hard tissue attachment, so we're looking at bone, we want to make sure that osteoblasts like the surface, and we assess that by looking at gene expression. Here you can see a sample gene expression. This is osteopontin. It's a marker that's related to osteoblast differentiation. And you can see an increase in differentiation on two perspective surfaces when compared to our machined titanium surface. When we run the in vitro assessments, we use just discs, 1/2 inch diameter typically, and this is how we run the cell proliferation, the cell attachment, and differentiation assays in the lab. If all the in vitro assessments go well, the next step is to look at an in vivo assessment. What we would do there is do an animal model. We look at reverse torque testing, and we take micro-CT, histology, and histomorphometry to analyze that reverse torque testing. For example, here you can see a dental implant, which we have done a canine transcortical model, and I'll actually show you a video here. So this is the micro-CT image 3D image of the Zimmer dental implant through this dog femur. If you have any questions, just stop and let me know. If you'll follow me, I will show you how we assess the hydrophilicity that I talked to you about. So here you can see a couple of perspective surfaces, and we'll be looking at them using the goniometer to assess the hydrophilicity. If you watch the screen, this is a live video feed. This is a polished titanium. So we would expect from the titanium is hydrophobic surface. So you'll see when this water droplet touches the surface, it's going to beat up. What we want, as I said before, is a hydrophilic surface, something that the water is going to spread on and therefore increase that cell attachment. So we're going to slide over to a perspective surface, and I will show you the hydrophilicity of this surface. So with this perspective surface, as soon as you see that water droplet contact the surface, you'll see an almost immediate spreading.
Here you go. Okay, next we will talk about trabecular metal or TM technology. TM is a unique material in that it has three-dimensional interconnected porous structure. So on this screen, we have two images. On the left side is trabecular bone image. On the right hand side is a TM image. Here we can see that the TM material have very similar structure as compared to the human trabecular bone. Another beauty of the TM material is that TM technology is versatile in making TM materials into different shapes. For example, we have a TM disc here, and also we have TM coated cup. So we also have the capacity of making TM materials into different porosities. For example, we have 3 different TM materials with different porosities here. This is 65 ppi, means pores per inch. And we have 80 ppi TM, which is the materials that are currently on market. And we can also make TM 100 ppi materials. You can clearly see that these different materials have different pores and porosities. We have also done some in vitro cell culture work on TM materials. First of all, we have done fibroblast culture on TM materials, and we have shown that TM can support fibroblast growth on the materials. You can see that from day 1 to day 10, cells can grow inside the pores of the TM materials. We have also done some in vitro cell culture work using human osteoblast cells on TM materials, and you can clearly see that at day 1 cells can attach to the TM surface very well, and at day 7 and day 21 cells have covered the majority of the surfaces, and also cells can grow inside the TM porous structures.